Hello my dear students, I hope all of you are doing very well and you are learning every day. My name is Abhijit Devnath. I am working as an assistant professor at NIT Pharmacy Institute in Greater Noida. Today I am going to tell you about eukaryotes and prokaryotes, about the genetic organization, that how genetically prokaryotes are different than eukaryotes, and with passing of time what are the advancement was happened in eukaryotes and how you can take the knowledge to apply in the field of biotechnology. So, I am going to tell you about all these things. First I will tell you what is prokaryote and eukaryotes, about prokaryotes genome, then I will tell you eukaryotes genome, then what are the differences, that is general differences, then in chromosomal differences, in DNA what are the differences in the DNA, and last I will end up with the summary. So, very interesting things we are going to learn. So, let me first with the picture. You can see here. I hope all of you have studied, all of you have drawn these pictures during your school days. Eukaryotes, this is very simple, and before starting the actual discussion, let me tell you few things. I hope all of you have studied about the evolution, that in evolution there was only single cell which was actually generated in the sea. We call it as a bacterial cell, and from that with passing of evolution that is a prokaryotic cell become eukaryote. A lot of advancement don't happen in eukaryote. Today we all human, we have these kinds of cells, eukaryotes, but our ancestors old ancestor, that is bacteria, they were actually carrying these kinds of cell. There are some similarities, despite the evolution there are also still some similarities are present. Now, let me show you what are the similarities. First, both the things are cell. Second, you can see in prokaryotes you will be having the cell wall, okay, then pilus, and then nucleate, and somewhere you will be getting some flagellum, and yes, these things are actually highly unique in prokaryotes: nucleates, pilus, cell wall and flagellum. In modern days or modern cell or our eukaryote cell, you will be not observing this kinds of thing, but few are the common things which are present in prokaryote and also in the eukaryotes. What are the things? Cytoplasm, okay, cytoplasm is the common, then ribosome, then plasma membrane, both the things are highly common, but here where these things are unique, in eukaryotic cells also few things are unique, like lysosome, then your mitochondrion, then endoplasmic reticulum, and also Golgi apparatus and nucleus. Okay. Here you can see nucleate, here is a nucleus. So, these are the few basic differences you can see in prokaryote and eukaryote. Another thing I like to tell you, that these things are highly unique in prokaryote, and for eukaryote all these things are highly unique in case of our eukaryote. So, these are the basic differences in prokaryote cell and eukaryote cell. Now, let me tell you something about the prokaryote genome. As because we are learning pharmaceutical biotechnology, we are more concerned on genome and how we can use this knowledge in our that is a pharmaceutical biotechnology. See, genome sequence data have identified indicated that most prokaryotic genomes consist of a single circular DNA molecule containing 580 kilobase pair to more than 4600 kilobase pair. Okay. So, you can see what is the range 580 to 4600, that is a number. Yeah. Then if I tell you more, the extrachromosomal plasmids or bacteria contain also may such elements known as the genetic bacteriophages. Okay, like various kinds of bacterial virus, that is bacteriophage T2, they are actually containing some extra chromosomal plasmid. Okay. Many of the bacteria contain additional plasmid that ranges in the size of several to 100 kilobase pair. Genes on plasmids are small genetic element that replicate independently on the bacterial chromosome, and yes, most plasmids are circular DNA and double stranded DNA molecules varying from 1500 to that is 4 lakh base pairs. See, I hope you remember my previous lecture where I have told you about the plasmid. Remember my lecture on rDNA, rDNA, where I have told you that we are actually producing recombinant DNA by using bacterial this thing called plasmid. So, this is the most important part about the prokaryote. Bacteria are actually prokaryote, but we can use their plasmid for our recombinant DNA technology. Now, we have seen this few knowledge. Now, let me tell you more. DNA circles or chromosomes or plasmid which contain genetic information necessary for their own replication called replicons. Because you see ultimately the genomic material, it will be translated, it will be transcribed, it will be translated, and due to that a new bacteria will be created. Okay, that is why it is necessary for their replication. Bacteria usually have only one copy of their chromosome. And if I tell more, because bacteria have only one chromosome, alteration of gene will have a more obvious effect on the cells. Yes, sometimes mutation are also happening, due to that some bacteria have become so much resistant to the various antibiotics. I will tell you more in my upcoming lecture what is antibiotic resistance. Then if I tell you more, many plasmids carry genes that mediate their transfer from one organism to another, as well as the gene associated with the acquisition of or requisition of the DNA. See, it is a very important. Let me make one star mark, because due to that nowadays we are observing AMR, antimicrobial resistance. I am going to tell you in detail about how antimicrobial resistance is happening due to plasmid. One of my lecture will be on conjugation, transduction, transformation. In that lecture I will tell you more how these things are actually creating a big problem to us. But yes, plasmids sometimes also play a positive role also. Therefore, the gene with independent evolutionary origin may be assimilated by plasmid that are widely disseminated among the bacterial population. Transposons are genetic elements that contain several kilobase pair of DNA, including information necessary for their organ that is a migration from one that is a genetic locus to other. Transposon, then your plasmid, we have a separate lecture on particular this topic. For today you may just learn these are some process carrying some information. These are carrying some information, and that information can pass from one bacteria to another, and due to that this bacteria, due to that this bacteria will be having much more property than this. Okay. Now, after learning prokaryotes, now let me start with the some discussion with the eukaryotes. So, what is the first feature of eukaryotes? The first feature is that multiple genome, okay, nuclear, that is a plastid, mitochondrial also one kind of plastid, chloroplast, multiple genome. Then plastid genome are responsible resemble prokaryotes genome. Multiple linear chromosomes you can also see, these are the number of genes or their that is a megabyte, how their size. Then monocystonic transcription unit, then dicontinuous coding regions, intron and exons. In our cell there are some region, in our gene there are some region where there will be non gene, where there will be non gene or such genes which will be not translated to create some protein. Okay. Large amount of non coding DNA, yes, that is a true fact for our human also. In our human genome there is a large number of that is a DNA which is actually that is a not coding any information to translate. This is we call it as a non coding DNA. Transcription and translation takes place in different compartment. All of you are aware that DNA to what will happen? mRNA, and from mRNA what will happen ultimately? Protein. Hai na? So, here transcription and here translation is happening, okay, DNA to mRNA to protein. And if I tell you more, variety of RNAs including like coding mRNA, rRNA, tRNA, non coding siRNA or snRNA are also playing a big role. Often diploid genomes and obligatory sexual reproduction is playing a big role here, and standard mechanism of recombination called meiosis. Okay. So, these are the some general features of our eukaryotic cells. Now, if I tell you some what are the general differences in terms of DNA, here DNA is naked, and here DNA in prokaryotes DNA is completely naked. Eukaryote DNA is bound to the protein. Then it is circular, here it is linear, usually no intron, and here usually we have intron, that is non coding gene. Organelles, no nucleus, we have one nucleus, then no membrane bound, or you have membrane. 70S ribosome, we have 80S ribosome. I hope all of you are aware that 50S, 30S, somebody also got Nobel Prize from India, I think in 2008, because he has worked on particular this area. In reproduction, there is binary fusion, we have meiosis and mitosis. Then cell single chromosome, it is haploid, for eukaryote it is pair diploid or more. What is the average size? Smaller, very small, it is larger. Okay. For for prokaryote 1 to 5 approximately in micrometer. In our case for eukaryote 10 to 100 micrometer. If I show you the picture, here is the picture, I hope already you have seen in the previous. Actually here I am trying to tell you in the context of chromosome, how they are different. See, this is the prokaryotes DNA nucleate, that is open, and in our case this is our nucleus. In that nucleus we have chromosome. If you take your chromosome and if you just unwind, you will be observing that chromosome actually having two things, one is DNA, another is histone protein. DNA is wrapped around the histone protein. If you just unwind all those things, you will be getting double helix like structure, you can see double helix like structure DNA. So, can you see how different our this eukaryote with our these things called prokaryote. If I tell you more, double stranded circular DNA in that is prokaryote, here long strand DNA containing gene information made up of a single chromosome, here it is containing several chromosome. I hope all of you are aware, we human having 23 pair, out of this 22 are autosome plus 1 is the that is a 1 pair is the sex chromosome. Okay. Then to occur in nucleate, and it occurs inside the nucleus. No homologous chromosomes can occur here. Often you can have a pairing of homologous chromosome. Attached to the cytoplasm of from inside have no cytoplasm. It is a short circular DNA. We have a long linear DNA molecule. You can see it is a long linear. Okay, you can see long. Here you can see it is very small. If I tell you more, lack of telomeres or centromere, we have telomeres and centromere associated with the nucleate or associated with the protein, it helps them for by packing. In this case, have special structure called nucleosomes along associated with the histone protein. We have seen in our previous picture what is histone protein. These are actually histone protein. Then contains several organ that is this thing called organs for replication per chromosomes, and in this case contains single origin of replication. If I tell you more, replication occurs at the beginning of the cell division, and here replication occurs in the S phase of the cell cycle. Okay. So, if I tell you more, originated in the operand, here it is regulated in individual structure and encodes for few protein, here it is encoded for large number of protein, and yes, achieve genetic recombination through horizontal gene transfer 1, 2, it is horizontal gene transfer, and here achieve genetic recombination through meiosis through fusion gametes. Okay. Basically during the preparation of the gametes, meiosis is actually happening, and that time only the gene recombination can be happen. In bacteria case, in case of bacteria, plasmids through transduction and translation it is sharing. Now, we have seen in chromosome level what are the differences. Now, let me tell you something about the in the level of DNA. So, prokaryote DNA actually found in the cytoplasm, prokaryote cells and circular plasmids. In our case, in prokaryote DNA is found in the nucleus of the cell inside of the chloroplast or mitochondria also, you can see. They are not found in organelles, and here some DNA found in the inside of the chloroplast or mitochondria. Then consist of one copy of genome, here consists of the more than one copy of the genome, because we are actually inherited something from our mother called mitochondrial that is a genome. Then contains small number of gene, we have a large number of gene. I hope all of you are aware how much that is how much big our genome is. Then organized in a single chromosome, we have a many number of chromosome, and it is also not packed in histone protein. We have, that is eukaryotes having a histone protein. This is circular, we have a linear, okay, we have a linear like this. Then consists of one copy of genome, we have more than one copy. Then contains a small number of gene, we have a large number of gene. Then organized into a single chromosome, we have organized in a many chromosome. Then introns are present, no intron, introns are actually absent here, and we have intron. Introns are basically non-coding gene. Introns are basically non-coding gene. DNA replication is occurs in cytoplasm. Here it is happening in the nucleus. Okay. See how different prokaryotes are with our eukaryotes. Then chromosome containing a single origin of replication. Here chromosome contains many origin of replication. DNA's replication is rapid. Here it is very slow. DNA replication is much more slow in case of us compared to prokaryotes. It consists transposons. We do not have any kinds of transposons. I will tell you more what is transposome in our transposons in our next lecture. Then genome, that is a GC content is more than 80. What is GC? I hope all of you have remembered these things called ATGC, adenine, thymine, guanine, cytosine. Okay. GC content, guanine and cytosine content is more than 80. Okay. In case of prokaryotes, and here T content, that is thymine content is more than that is a 4x of GC content. Okay. Says that is the size of the DNA is very less, and here in case of us our size is much more higher. Now, in summary, because we have discussed a lot of things in particular this topic, see another thing I have to tell you apart from our this learning. You see, with passing of time there are lots of advancement, or I can see evaluation is happening due to mutation, and due to that bacteria are changing themselves, and bacteria are actually our actual ancestors. From them there are lots of evolution happen, whatever you have studied in the chapter of evolution during your school days, from one cell to bacteria, bacteria to some that is animal, some that is a species in that is a sea. After that fish, from fish to various kinds of amphibian, then reptile, then birds, then mammal. So, a long story from prokaryote to eukaryote journey. Prokaryotes are our main ancestors, the bacteria. Okay. So, in summary, if I tell you, so prokaryotes are actually nonsense genome, nearly all coding, okay, no nonsense genome. Whatever the genetic material is present in case of prokaryotes, all the genetic material it will definitely transcribed or translated. In our case, we have a lots of non coding genome, we call it as an intron. Frequent horizontal gene transfer HGT means if you have one bacteria, it has some plasmid, here is another bacteria, they can share the information through sharing the plasmid, that is, and it will get the information from this bacteria. We call it as a frequent horizontal gene transfer HGT. In our case, less frequent HGT, there are for human it is very impossible or very less. Circular genome, circular genome plus plasmid, we have a distinct linear chromosome. Operons are actually present and more distance in regulation. Streamlined genome, streamlined genome, and this case also that is a few repetitive element, and in this case, in our case, abundant repetitive element. Same gene, it is present in repetitive element. Okay. Very rapidly evolving in both sequence and structure and more conservative mode of evolution. Very large population size up to 10 to the power 9 to 10 to the power 10, and here the most interesting thing is that smaller population size. Okay. Population size is very small for the case of eukaryotes, because they are evolving, they already have evolved and they are the advanced version. Most important thing is that their size, the organism size is much more bigger than prokaryote. Prokaryote is basically only one cell, you can say, bacteria is only one cell. So, that is why they are very large population, present in the world, and we have a very small population. So, today we have seen that what is prokaryote, what is eukaryote. You also have learned that about the prokaryote genome, where I have covered that what are the features of their genome, how they are assembled together, how they are actually that is available in the in its cell. And you also have seen in that prokaryote genome there is no nucleus. It is very interesting. Na? There is no any nucleus is present in the prokaryote the genome, and most importantly whatever the prokaryote genomes are present, it can entire part, there is no any intron, there is no any intron. Whatever the genetic material actually present here, everything will be transcribed. Let me erase, yeah, whatever the genetic material is present here, this thing will be transcribed into a mRNA, and then it will be translated translated into protein, sorry, translated will be written here into protein, okay, forget this thing. So, entire part whatever the genetic material is present here, all the parts will be transcribed and translated, but in case of eukaryote the story is different. Lots of introns are actually present, and due to that all the parts will be not properly get translated, that is transcribed. Another thing I will tell you, that in the 21st century all of you might be thinking that sir, we have studied the same concept or same thing in a very that is a simple manner during our school days. We have studied about that lots of organism, lots of that is cell organelles, like let me go to the my first slide. Yeah, here all of you might be thinking that sir, in our school days we have studied about only these things, that these organelles are actually present in eukaryote cell, and these organelles are actually present in prokaryote cell. But today we have got to know how genetics actually associated with this. You see, in my entire lecture I have told several times that how is genetic material actually assembled. Why we should learn? We should learn because this is a very simple organism. This is a very simple organism. Due to its simplicity, entire genetic material it will be translated and it will create some protein. Due to that, we are actually using bacteria in a very rapid manner in our biotechnological field, like producing of insulin, producing of various antibiotic, or in vaccine production also. We are actually using those organisms, because entire genetic part actually coming to translate. But in this case, due to presence of lots of your intron and other non coding gene, it is problematic. Okay. So, yeah, so that is why this is actually playing a big role. I hope you are actually right now so much you have a very clear concept with the prokaryote. In eukaryote you have seen that it is a very big, it is a very large and huge amounts of information is present in the eukaryotes, and due to that there is a huge diversity. I have also covered what are the difference between the prokaryote in terms of general, in terms of chromosome, in terms of DNA, and lastly I have ended with the summary. I hope all of you have enjoyed this lecture. In next lecture, I am going to tell you something very important thing. I will tell you about the microbial genetics. We are going to enter in the much more deeper concept, how transformation is happening, how transduction is happening, how conjugation is happening, plasmids and also transposome. In my at the beginning of my lecture several times I have mentioned that one bacteria is present here, here is another bacteria, this bacteria having some plasmid. So, it will be able to communicate with this, and plasmid can be shared, and this is will be called as a SGT. So, due to SGT this bacteria will having much more information, information compared to this. So, that is why how these things are happening, how transformation and transduction is playing a big role to create a big problem called AMR, antimicrobial resistance, or we sometimes call it as a drug resistance. So, all these things I am going to tell you in my next lecture on tomorrow. So, thank you all of you for joining today's lecture. I hope all of you have enjoyed the entire lecture and you got huge information which information will help you in your studies, and in next lecture we are going to learn something more. So, thank you all of you for joining. All the best. God bless you all. We may meet in our next lecture.